So in the last class, we spoke about mutagenesis, right? We spoke about mutagenesis, and mutagenesis is all about changing the alphabets of DNA, nucleotide ATGC. But what is the consequence of this mutagenesis? The consequence is protein. Because DNA, as you know, central dogma, DNA transcribes to RNA and RNA to protein. So protein becomes the major workforce in a cell. Now, when we want protein to be made at an industrial scale, our cell is very small body, small sac, and there are thousands of protein which are produced in very minute amount, in a very less amount. So if you give the same condition, it may not produce industrial scale protein. You want industrial scale protein for therapeutics or for diagnostics or several other things, but these are the major on which the biotech industry is counting on. Industrial scale production of protein, and protein is just one of the biomolecules. We will look into RNA and in next classes, right? So we are going from mutagenesis to proteins, and to produce cellular protein in an artificial system, we need different models, like bacteria, and so forth. So it's possible that the protein that is produced, the human cellular protein that is produced in an artificial system like bacteria or yeast, then it has to be modified, and then this protein becomes a recombinant protein, because recombinant protein, because of the genetic identity, right? They're produced either through using plasmid or other kind of vectors. So it becomes a recombinant DNA. So the protein also is a recombinant protein. So today's chapter is focused on recombinant protein, and that's the rationale for this chapter. And I have put this chapter in one folder on the blackboard so that you can study it together. So going to my slides. So let's say, what is a protein, or what proteins are? So they are biological macromolecules, biomolecules, and they're very active in terms of doing a function. So what function a protein can do? So protein can do many functions. And some of the listed functions are proteins that can catalyze metabolic reaction. Metabolism is center to generation of energy from food particle, right? And that involves several system, digestive system, and then in mitochondria, oxidative phosphorylation, glycolysis. The whole biochemical pathway is dedicated to metabolism, and metabolism is like cascade. One events lead to other, and the whole lies at the back of the protein molecules, enzymes mainly. So, and then proteins that are involved in replication, transcription, translation. Even ribosome, if you see, ribosome is a complex of RNA and protein. Then proteins also help a cell to respond to to stimuli. Now, why I say this? I'm saying this because let's consider that this is a cell, and this is a nucleus, and within the nucleus there is DNA. And if there are any changes that happens in the extracellular environment of a cell, or let's say if a cell is hit by a UV light, then cell passes this signal in a stepwise fashion to the nucleus, and nucleus to the DNA. And then DNA, it makes RNA, and RNA goes to the cytoplasm, and then protein is made, and then this helps the cell to respond to various extracellular changes. It could be viral infection, it could be exposure to UV light, it could be hormonal changes, right? So proteins, they communicate outside changes happening in the cell to inside, and then takes the message to the nucleus in the DNA, and then DNA prepares the program within the cell to respond to these changes. This could be the backbone for infection. This could be backbone for immunity. This could be backbone for hormones. This could be backbone for many processes, signal transduction. Then transport molecules. Proteins, they form transmembrane. So our membranes, they're bilipid layers, and within this bilipid layer there is mosaic of proteins, and these proteins could be integral, transmembrane. These proteins help. So protein is everywhere, and any deficiency in the function of a protein can cause the cell to go erratic, and that can become a nucleus, a beginning of a disease process. Right? For example, let's say transmembrane proteins. Transmembrane proteins are integral proteins within mitochondria. They help in the generation of ATP, ATP synthetase, oxidative phosphorylation pathway. If there's a problem, then ATP is not generated. If ATP is not generated, there is no energy in the body. A person cannot survive. So a single protein can have huge impact on an organism, including human being. So this is about proteins. So there are, in majority, there are three forms of protein. Alpha helix, right? The name itself suggests that, helix shape, but this is not double helix like DNA. DNA is single helix. What is the property of helix? The property of helix is that, let's say you, for the sake of understanding, let's say you draw a line here which is straight, right? So the quality of a helix, or the property of a helix, is that it will use this line as a central, and it will go around like so. This line is in center, and the protein ribbon will curve around this hypothetical central line. And how this is stabilized? The alpha helix is stabilized by hydrogen bond between NH amino group and carbonyl group of fourth amino acid. This is very important. Alpha helix, it curves around a perpendicular. And if what you see here, that whenever, so there's a geometry here, whenever there's a turn in the helix rises. So if you see here, if you notice these things, the helix rises at every turn. So again, the helix would rise here. Helix rise here. So there is a dimension here, and this happens around a central perpendicular structure. And peptide and alpha helix is stabilized by hydrogen bond every fourth amino acid. The second category of structure is called as beta sheet. Beta sheet, the hydrogen bonds are formed laterally. So there could be three beta strand, right, in this situation. So alpha helix, beta sheet. The third form of structure is coiled or random coiled. So random coils refer to these structures. A random coil refers to a string of protein which is neither alpha helix or beta sheet. Because of the sequence of amino acid, they don't form alpha helix or beta sheet. So this alpha helix is formed because of the sequence of amino acid. Alpha helix or beta sheet is also formed because of the type of amino acids that are there. And if a protein doesn't have that kind of amino acid, then this is coiled. Generally, coiled is used to connect two secondary structure. It could be two beta sheet, it could be two alpha helix or alpha helix or beta sheet. Now, because of this combination, permutation and combination of alpha helix, beta sheet, coil, the proteins, they can adopt different structure. Here is, you see, this structure is just like a donut, Dunkin' Donuts, right? There is a hole inside and then there is a spherical thing inside. So most likely this protein is used for channels inside the cell membrane, so that flow of ions through this protein. Then protein so within a lipid layer, protein could be inside, inner protein. There could be an external protein. There could be protein that could be just inside this lipid layer, and there could be a protein that could, transmembrane protein. It goes through the membrane. Cytoplasmic tail in the inside of the cell, amino terminal on the extracellular space. Transmembrane protein, they are generally protein receptors. This could be ion channels. Then some proteins, so if you see here this totally beta sheet, this could be cellular receptor. Cellular receptors like epidermal growth factor and so on and so forth, cathedrins. So number one, proteins have many functions: enzyme, cytoskeletal, muscular, transcription, translation, ion channels, hormones, major workforce. And this protein, they have a secondary structure depending upon their orientation in the three-dimensional space. Now, building block for a protein is amino acid. And I mentioned to you in a couple of classes previously that there are 20 amino acids. These 20 amino acids can be distinguished from each other by side chain, and that is R. Nevertheless, there are 4 groups of amino acids: non-polar, polar, acidic, and basic. So basic amino acids are positively charged. So, histones, we talked about chromosomes in the last class. So chromosomes they are made up of chromatin, which is made up of DNA and protein. Proteins are positively charged, and these positively charged amino acids are lysine, arginine, and histidine. They compose histone. That's why the interaction between histone and DNA is very tight. And this tightness helps the DNA to be packaged. Otherwise, DNA is multi-meters long, and it has to be spaced inside the nucleus So it has to be superly packed. So these positively charged amino acids within the histones, they contribute. Negatively charged amino acids are aspartic acid and glutamic acid. So they have carboxyl moiety in their side chains, and they help in forming ionic um, electrostatic interactions or hydrogen bond. Polar amino acids, hydrophilic, that likes water, but they're non-jar. Glycine, serine, threonine, cysteine, tyrosine. The one thing you notice that these hydrophilic amino acid, particularly serine, threonine, tyrosine, they undergo phosphorylation. These are the three major amino acids that undergo phosphorylation. Phosphorylation plays an important role in protein modifications and signal transduction. Hydrophobic residues. Hydrophobic residues, as the name suggests, they don't like water. So they are not on surface, right? So protein can be hypothetically, for sake of explaining, one can say that if this is the three-dimensional structure of a protein, then on the surface, generally, there are charged amino acid, right? This helps this protein to solubilize in water, right? Our cells, 70% of the cell is water buffer. So this helps the protein to stabilize. But what helps the protein to fold, right? Protein is folded in three-dimensional space. This folding is achieved by hydrophobic amino acid. So they are not on the surface, they are inside. They help the protein to fold, right? And folding is important for protein to give conformation and function. So there are, you can read these write-ups in detail. The chemical interactions that stabilizes protein structure is hydrogen bond, as I mentioned, in alpha helix and beta sheet. Then hydrophobic interaction helps the protein to fold, right, to become stable. Then there are electrostatic interactions, salt bridges, right? And as our side chains, positively charged or negatively charged amino acids are involved here. Then the weakest is van der Waals interaction. So all these interactions, they work together to stabilize a protein. Protein has immense function inside our cells, right? So thing is, why do we need a recombinant protein? As I mentioned in the beginning of the class, therapeutic, diagnostics, and to study basic science or some other important value. So if you see this figure here, this is a figure to show the role of protein in drug discovery, in discovery of new medicine. So we can use three model systems using proteins. We can use three model system to discover new medicine. First model is we can directly use animals or fly model. This is very risky, and there's chances of failure is more because it's very hard to know what is happening inside the mice body. But it is very important because if a medicine is working inside, if a new medicine, new molecule, potential medicine is working inside mouse body, it's a good sign that it could work in mammals also, in humans also. So here, let's say this mice is a disease model for let's say malaria, and you know that malaria could cause brain damage or fever. So to discover new, and malaria, we don't have vaccines. So if you're discovering malaria, anti-malarial compounds, then you make this mouse as a malarial mouse, and then you want to test various drugs, and these drugs will act on the parasite of malaria, Plasmodium, whatever the model is, Plasmodium falciparum or Vivax. So it is a model to study new medicine. The second model is just to make a recombinant protein. Pay attention. Make a recombinant protein for against which you want to make a drug. So let's say there is a protein inside a malarial parasite called as cysteine proteases. And these proteases are important for evading the immune response. So what you will do, you will get the cysteine proteases from malarial parasite and then see which molecules can inhibit the activity of cysteine proteases in malaria. And why we cannot use malaria? Because it is very hard to grow malarial parasite in labs. So best is to clone the gene for cysteine proteases and make abundant of protein. And these proteins can be used in various assay systems to discover new molecule. So recombinant protein. Similarly, recombinant protein can be used for more focused structure-based drug design, where you know the structure of a protein and you understand which amino acids are involved in really binding so that you can make the drug more potent. So structure-based drug design. So all these are based upon protein. This is, you don't know here the real target because these molecules will go inside mice and you don't know which protein they are acting. Although you know that, okay, this molecule is acting against malaria because mice is getting healthy after ingesting or taking up these drugs, but you don't know which protein is involved. But here, because of recombinant protein, the power of recombinant protein for generating recombinant protein, you can directly see direct target-based drug discovery or discovery of new medicines. So, so I just, we talked about this in the class that chromosomes are made up of chromatin, and chromatin is made up of nucleosomes, and each nucleosome is made up of histone proteins, which are octamers, which is dimer of H2A, H2B, H3, and H4. So octamer, and these octamer histone proteins, they're called as nucleosomes, and between nucleosomes there is DNA and also histone H1. So they also because these they are controlled. The structure of DNA or structure of chromosomes is controlled by nucleosomes, and on the nucleosomes, various proteins can function by modifications like methylation or acetylation. So there are many medicines that can be made directing at methylation. As I mentioned to you, the example of EZH2, which is methylating a target histone H3 on lysine 27. So in case of prostate cancer, it can be. So these are, little bit. So this is a normal prostate, but in prostate cancer, the size of prostate increases dramatically. And here EZH2 could be mutant, which may not be doing its function of methylation. So the new molecules can be generated against EZH2, or you remember I was talking about the production of a protein from a paramecium virus, chlorella virus called as PBCB1, which also express EZH2 like protein, but only that can methylate histone H3 at lysine 27. So even that protein, set protein that mimics EZH2 function has to be made in abundant, right? So that it can be trial, because a protein has to go under various trial to finally reach to a pharmacy. So you need large amount of protein. So this has to be done. Anything that is that has to be scaled up, right? So let's say if you want to go for an MCAT or nursing exam or PA exam, then you have to program yourself. The more you program, as the better chance of success, right? So this recombinant DNA technology also is very, very planned. So right from you have to choose very carefully, very smartly, which proteins to choose from 20,000 proteins that can be good for making a product or publishing a good paper in basic science. So you have to choose the right plasmid, right mammalian cell. If it is getting glycosylated, then you have to use insect cells. Then you have to purify the protein. You have to do the quality control, package it, and then deliver. So these proteins can be used, for example, restriction enzymes, right? They're used in molecular biology lab. So they are research tools. Then various reagents. Um, for example, you have to use markers for doing chromatography. You need proteins, various assay, enzymes, therapy. So you need proteins for all these purposes. So you have to plan. This is called as platform-based science. From one platform, you go to another, to third, and this is how you control the process. If you don't control the process, then it will go in all direction. Your company will flop. So you have to do a platform-based approach to for a biotech company if you're making a product. Right? So when I'm teaching also, I try to do it in a way, in an organized way, such that I can be more, I can explain myself in the best situation. So protein, which is a target here, can be made or maybe expressed in E. coli, right? E. coli has its disadvantage and advantage. Yeast system, if a protein needs to undergo post-translational modification. Baculoviruses, for those proteins which are heavily glycosylated, like I'll come to that. Zona pellucida. So if you want to develop a fertility vaccine, if you want to break sperm and egg interaction, either you have to develop vaccine against sperm or vaccine against egg. An egg is covered by a huge, a very complex jelly-like uh, glycoprotein called as zona pellucida. So that can be expressed using baculovirus system and then the mammalian system. So you have to see which system is good for your proteins, recombinant protein for expression. So this was the example, but I hope you remember my from the last lecture or last to last lecture, I almost dedicated last two lectures to proteins, where a protein can be expressed in a vector system for accelerating the production of the protein so that it can be used. So baculovirus is that system that is used to express heavily glycosylated protein zona pellucida. So what happens that there was a time when government were spending a lot of money on contraceptive strategies. So one of the strategy was the first strategy was, so let's say this is a sperm, right? Then scientists figured out that there is a protein which is present here that binds to egg. So if we develop an antibody against this protein, then the sperm will not fertilize with egg. Right? The second strategy was that human egg is covered by a protein called as zona pellucida. Zona pellucida, which is a glycosylated protein, and it was found that there are three or four, but as much as I know, it is zeta pellucida 1, ZP2, ZP3, and out of which it was found that ZP3 or ZP3 has a sperm binding site. So if you make antibodies against, if you block ZP3 here interaction with sperm, then they will not fertilize. So this molecule or this antibody can be a potential contraceptive vaccines. So these are we have to make zona pellucida or sperm glycoprotein in tons of amount so that we can do various biological assay to make sure that this protein is important for the fusion of sperm and egg. So if the function of this protein is blocked, then it could serve as an effective contraceptive vaccine to control the population in various parts of the world. So at some point of time, a lot of countries that took initiative in this direction. So here you have to choose, if you're using, if you're trying to use zona pellucida, then you use baculovirus system, right? Baculovirus system to similarly, if there's a protein that goes post-translational modification, then you can use yeast system. You may not have to use a bacterial system because bacterial post-translational modification and humans are very different. So what is the platform-based approach for recombinant protein? You have to choose right expression vector for your protein. You have to use the right plasmid for this protein so that your protein is expressed. Then this plasmid can be shuttled inside the cell using chemical treatment. That process is called as transfection. And these cells carrying plasmid can be selected for those cells which only carry plasmid. And this then can be seen that which colony of cells express maximum proteins. And then you can scale up the cells and then get the cell, large amount of protein for various assay systems. Right? So, and then a lot of companies, they are now making, for example, let's say I'm talking about myself, like I got COVID, number one, then I got first dose, then I got second dose, then I got third. So that I'm already tired of taking vaccines, injecting needle all the time. And who knows, in future you may have to get booster, or COVID-19 can go the flu way that every season you have to take this vaccine. So many scientists are toying with this idea to make edible vaccines, right? So they can take the COVID protein, put it in banana or apple plant, and then so first they will have to find the right vector like plasmid, and that can take up COVID-19 cDNA or RNA, and then it is injected into plant cells, and this plant or fruit can express COVID vaccine. So or flu vaccine that we take every season. So how many times of needle piercing? So these are the approach that biotechnology companies are already working to have edible vaccines. But one of the challenge with edible vaccine is that it has to go through the stomach, and the pH of our stomach is very, very acidic one, in which many proteins may not be. Generally, these proteins, they are stable at pH 7 or 7.4 because that's the physiological pH. But the pH in our stomach is very, very acidic one. So many proteins can get cut off, chopped off there. So there are a lot of avenues to take, but there are a lot of challenges in each path. So that's how the technology comes in. It has to be improved all the time. So this is also one of the nice figure to say how to make a recombinant protein. So first you should know which protein that you're interested in for your project, for your company. Then you clone this, you sequence the DNA, the sequence of the clone, so that there is no mutation. So what if you're growing a protein, a hormone, and you did not sequence the DNA, and you have the mutant form of the protein, your whole project will be gone. So then you express this protein inside the cell or bacteria. Make sure that the protein is good through various techniques like mass spec, SDS gel, and then do all the quality control experiment, and then see the activity of protein, and then scale up. And then you're in business. You make one protein that can treat one disease, you can make so much of money. And not only money is just one aspect, but you can employ so many people. And when a company is formed, it changes the neighborhood, it changes the city, changes the state. And all it starts from is one idea. So, it can, the protein business or protein study or a function of a protein can be good for basic biomedical research to understand, because many functions we don't understand still, like at the level of, we may know one function of a protein, but proteins, they work as a network. So we don't understand many functions of a protein. So it can be used for basic biomedical research. It can be used for clinical research, such as epidemiology, then animal models, and scientific medical knowledge. So I wanted to finish this class in 35 minutes. So it's 5:33, so we are almost on time. This was a short chapter, and I intentionally did that so that the rest 20 minutes you can take your quiz, so that the quiz don't become as a extra time for you. So it is within the class time. Whether you take quiz at 7 a.m., 7 p.m. or 11 p.m., that doesn't matter. But this 20 minutes, I'm saving it for you, that you can use it for your quiz time. So with this, I would like to end this class. Any questions?
Professor, did you get me? I came in later. It's
Yeah,
Tia
you
Foster.
tell me your name, because
Tia Foster.
Tia Foster. Okay. Yeah, okay, I got you.
Okay.
Hi, Professor Ruhama Safran. I came in a few minutes late.
All right, no problem.
Thank you.
I saw also a couple of other people.
Professor, I also came in 10 minutes late.
Name.
Kayla.
Hmm?
Kayla, Cayla.
Yes, I saw you.
Okay, perfect. Thank
Bernard as well? Me too, Professor Anderson.
Yeah, I got you.
Got me, Professor Bernard?
Yes, I got you.
Thank you.
So if there is no other question, then may I end the class? Good luck with your quiz and stay well.
Thank you, Professor.
You're welcome. Bye.